You the same one who warned me last time? Thanks for doing that. When I'm not feeling well, I'm definitely flaking out, and I was not aware that I was not recording. I'm now recording. All right, can you see the screen?
Yes.
Thank you. So we talked about the bacteria, the mycoplasmas, and the mycobacterium are two families of bacteria that have atypical cell walls. The mycoplasmas, actually, they don't have a cell wall. And the mycobacteria, they have a very unusual cell wall that's very lipid rich. It has macolic acid in it, giving the cell wall different properties. The archaea also have different cell walls. The main difference is their cell walls do not have peptidoglycan. They have pseudomurine. It's another disaccharide. You can damage the prokaryotic cell walls. That will make them susceptible to osmotic lysis. You could damage the cell walls by using penicillin G, an antibiotic, and we'll talk a little bit more about that later. But penicillin G, and actually all members of the penicillin family, inhibit the peptide bridges in the peptidoglycan. And that will create holes in the peptidoglycan, meaning the peptidoglycan will still have the, what do you call them, the long lengths of peptidoglycan, but they won't be cross-linked as well as linked this way. And that'll create holes. There'll be a gap there, and then the cell membrane can protrude out of the hole. You can also damage the cell wall by using the enzyme lysozyme. It digests the disaccharide in the peptidoglycan. And there's different terms which you don't need to know for a gram-positive cell and a gram-negative cell. But lysozyme is an enzyme that your body makes, particularly the skin, as well as your tears of your eye, to help the body defend itself against bacteria. So the body makes this enzyme to help defend against bacteria. And actually, lysozyme is one of the enzymes in lysosomes. And that's why that name is so similar, lysosome and lysozyme, because lysosome has the enzyme lysozyme in it. Any questions about any of that? Gram-negative bacteria are not as susceptible as gram-positive bacteria, particularly to penicillin G. And it's due to the presence of the outer membrane of the gram-negative cell wall. And penicillin G cannot get through the outer membrane. However, gram-negative bacteria are susceptible to other antibiotics that can penetrate the outer membrane. An example is streptomycin. I'm mentioning streptomycin just because it's an enzyme, sorry, not an enzyme. It's an antibiotic which is can go right through the lipid of the outer membrane. So streptomycin is a lipid loving antibiotic. We'll just word it that way. Any question about any of that? Some antibiotics like penicillin G only work on gram-positive cells, And there are some antibiotics that only work on gram-negative cells. You don't need to know this, but oh, I thought I had it. Now it slipped my mind. Something B. Hopefully it'll come back to me, but there's an antibiotic that puts holes in the cell membrane of the cell or the outer membrane of the cell. And because of that, this antibiotic is particularly lethal against gram-negative cells. And for the life of me, I can't remember the name of that antibiotic. It'll come to me, just give me a minute. But the point is that there are some antibiotics that work on gram-positives, others that work on gram-negatives. Most antibiotics actually work on both gram-positives and gram-negatives. And penicillin G, we've mentioned many times already, and we'll talk more about it later, works on gram-positives. But ampicillin, which is one of the early members of the family of penicillin, is an antibiotic that works on both gram-negatives and gram-positives. Any question about any of that? All right, if not, let's turn to the plasma membrane. It is a phospholipid bilayer. We've already stated that, although it is more than just phospholipids. There are proteins in it, and there are carbohydrates in it, especially the eukaryote cell membrane, but the prokaryotic cell membrane has the same. There are different proteins. There are peripheral proteins, and that is a protein that is primarily on one side of the membrane or the other side. So external to the cell membrane or internal to the cell membrane. There are integral proteins and transmembrane proteins. And both the integral proteins and the transmembrane proteins span the entire membrane. The most important protein that we'll probably talk about this term are the transport proteins. And in the plasma membrane, the transport proteins obviously span the membrane, and they move one molecule either from inside the cell to outside, or they move it from outside the cell to inside. And the transport proteins are highly specific, meaning generally speaking, they move one specific molecule. Like we have a transport protein to move glucose into the cell. And we have another transport protein to move the sugar fructose into the cell. Any question about any of that? All right. Let's blow this up just a little bit. There's the phospholipids. And here you can see the phospholipids. So it's a bilayer, a lipid bilayer. And it's this fatty acid bilayer that acts as the barrier, preventing molecules from coming into or leaving the cell membrane. And molecules that we want to enter the cell or to leave the cell, we have a transport protein to move that molecule across the cell membrane. The fluid mosaic model of the plasma membrane states that the membrane is a mosaic of phospholipids and proteins, and that the membrane is as fluid as olive oil. Do you see my hand here? I can't see anybody, so I'm going to assume somebody's.
Yeah, we can.
Great.
Yes.
It's an animal hand, right? So you know that the only thing, I don't know what's a word, the only thing that is there is the cell membrane between the cells. And that's actually a fluid. And it's always amazing to me to think about that my hand or any other part of my skin is held together with a membrane, which is as fluid as olive oil. And if you know what olive oil is like, it's pretty liquid, and it's pretty amazing that my hand is held together by olive oil or something as fluid as olive oil. Olive oil, if you don't know, is a liquid at room temperature. But if you were to refrigerate it, it would become a solid. The proteins can move about in the fluid to function. And those of you who have had A and P, you do know that when a ligand binds to a receptor, which we're going to talk about it as being a protein, the proteins bring the receptor all together as the protein moves, and then the ligands will be brought into the cell. So the point is that some proteins can move in the cell membrane, and they're free to move wherever they want to go. That's not true of all proteins. Let me blow this up a little bit. Like this, probably a transport protein. It's not labeled that, but there's a pore there, so maybe it's a channel protein, which is essentially the same thing. It's free to move around in the cell membrane. But a protein, and there isn't really one shown here, so let's go ahead and put it right here. A protein which is anchored to either the intracellular matrix or to the extracellular matrix, it's not free to move about in the cell membrane. So some proteins are fixed in the cell membrane, and it's pretty easy to distinguish those because they're anchored not by the cell membrane, but they're anchored by being attached to either the extracellular matrix, and that could be something like bone. So think of it as a cell in your bone, and the protein here could be attached to the bone, and then that protein's not going to move. Or it could be anchored to the intracellular matrix, which is we'll talk a little bit about that. That's the framework of proteins running throughout the cell, and that sort of makes a skeleton, like the backbone of the cell. It's a skeleton, I'll word it that way. We'll talk a little bit about that later. Anyways, those proteins are not free to move, but all other proteins can move about pretty much wherever they want in the cell membrane. And the phospholipids are free to move about also. They definitely rotate around right here, and then they can move around this way and that way. Any question about that? That's actually how water gets through the lipid bilayer. And that is water can't get through. Let me blow this up. Water cannot get through the fatty acid portion of the lipid bilayer. And the water has to get through two layers in a bilayer. what happens is that one of the phospholipids moves, or the fatty acid phospholipids move this way, and then a water sneaks in where there's a gap. And it has to wait right here because it can't get through the fatty acid upper layer, but it just waits because while the water is there, the phospholipid bilayer won't move in that region and then throw out the water because lipids and water don't like being around each other. And what the water does is just waits right here until eventually a phospholipid moves away, opening up a hole for the water to get inside or outside the cell. I guess I'm doing it from inside to outside. Okay, everyone clear on that? Any questions about that? Okay, thank you. That's actually very helpful when somebody lets me know. All right, the plasma membrane shows selective permeability. This means it allows the passage of some molecules, but not the passage of others. Generally speaking, most molecules are not allowed to pass through the lipid bilayer of the plasma membrane. And the plasma membrane and the cell membrane are the same, if you haven't figured that out. No large molecules can move through the cell membrane without a transport protein. Ions have a positive charge, and they are repulsed from the lipid bilayer of the cell membrane. So they either do not move across the cell membrane, or they move extremely slowly. And how they move an ion is that the ion will be dissolved in the water. And as the water slowly moves to this position, waiting for this position to open up, it can bring in a little bit of ions as the water moves into or out of the cell. It can transport ions, but essentially very slowly do the ions move, or no ions move. Lipid molecules, on the other hand, can move right through the cell membrane. The lipid bilayer of the cell membrane is lipid, and other lipids can move right through it. But if you think about it, cells are mostly going to be around water, meaning your cells. I don't know if we can say that about bacteria. Yeah, most bacteria will be mostly in an environment rich in water. Your cells are in an environment rich in water, meaning you're mostly water. And so there aren't that many lipids that will come into contact with the cell membrane. But if the lipid does come into contact with the cell membrane, it can come into the cell. Some examples would be if you're eating a cholesterol-rich diet, or you're eating avocado, you're going to get lipids. And those can go right through the cell membrane. Now, the avocado won't be a problem for you, but if you get too much cholesterol, that could be a problem. Small molecules can move through the lipid bilayer, especially small nonpolar molecules like oxygen and carbon dioxide are the most important. Small molecules, nonpolar. they can diffuse right across the cell membrane. And to the cell, this is important because we want oxygen to diffuse into the cell and carbon dioxide to diffuse out of the cell. So those two molecules have to cross the cell membrane. Any question about any of that? All right, not hearing any questions, so let me move on. The cell membrane, particularly in bacteria, have some, what do I want to call it, special properties, special things that we need to talk about. I'm not sure how to word that. Yeah, I've only got it as miscellaneous here, so it's not very helpful. For example, the cell membrane of bacteria contains the enzyme for making ATP. So the enzymes involved in ATP production are in the cell membrane of bacteria. That is not true for eukaryotic cells. Eukaryotes have the enzyme for making ATP in the mitochondria. For the photosynthetic bacteria, all of the pigments involved in photosynthesis are found in foldings of the cell membrane. And you don't really need to know the names of the foldings, but they're called chromatophores or thylakoids. And so we can blow this up. That's actually a folding of the cell membrane. So the cell membrane comes around right here, then folds in, circles, and then comes out. I'm not really drawing that too well, so hopefully you understood what I was trying to do. This is only found in the photosynthetic bacteria, the enzymes and the pigments for photosynthesis found in the cell membrane. In eukaryotes, the pigments for photosynthesis are found in the, losing it. Boy, I'm having a hard time today. Sorry, I'm not feeling well and I'm losing it. What is the organelle in plants that engage in photosynthesis? Come on, somebody's got to know.
Chloroplast?
Yes, chloroplast, thank you. Yeah, the chloroplast has the pigments for photosynthesis. They're not found in the cell membrane of eukaryotes. They're found in the chloroplast. You can damage the plasma membrane, and if you do, you'll cause leakage of the cell contents, meaning if you damage the cell membrane, regardless of how you damage it, but let's say you put a hole in it, then the cytoplasm will leak out, and that will kill the cell. You can damage the cell membrane with alcohols, quaternary ammonium compounds, and polymyxin antibiotics. Remember, I was trying to tell you the antibiotic that damages the cell membrane or damages, or what was I saying, that it could go through lipid bilayer? It's polymyxin B, that antibiotic. My voice might not hold out today. let's talk a little bit about movement of materials across the cell membrane. The movement of materials across the plasma membrane is critical to cells. There are two processes used by cells to move molecules either into the cell or out of the cell. There's a passive process where the molecule moves down a concentration gradient, meaning the molecule moves from areas of high concentration to areas of low concentration. I hope you didn't get to see that. There are three passive processes. There's simple diffusion, and that is where a solid moves from areas of high concentration to areas of low concentration. And we have an example of that, and that is oxygen moves into the cell by simple diffusion. And oxygen is dissolved in the air, or if you're in the pool, I guess, well, not really. For your skin, oxygen could dissolve into the skin from the pool water. And the point is, I'm trying to say is oxygen is a solute either in the air or the water. There's also facilitated diffusion. And this, the molecule does the same as in simple diffusion. It's just facilitated diffusion has the molecule moving with some help. And first of all, the molecule is moving across a membrane, such as the cell membrane. And the molecule is being moved or facilitated by a protein, such as a transport protein. Any questions about that? A good example would be the sugar fructose moves into the cell by a passive process, and it moves into the cell through a protein, which facilitates the fructose moving into the cell. And obviously it's moving across the cell membrane. This also involves a solute, meaning the fructose is dissolved in a solution outside the cell, probably water. Any question about any of that? The third passive process is osmosis. And osmosis is the movement of a solvent across a membrane. Osmosis is so critical for life that we have sort of a separate term here. And we're just going to call it osmosis because water is important to life. And it's a little different in that it's a solvent instead of a solute. And whenever water moves across a membrane, from areas of high concentration to areas of low concentration, it is moving by osmosis. Any question about any of that? All right. The second process used to move materials across the cell membrane is an active process. An active process is where the molecule moves against its concentration gradient, meaning the molecule moves from areas of low concentration to areas of higher concentration. So the molecule is moving against its concentration gradient. How this happens is the cell uses a transport protein. And to move the molecule from areas of low concentration to areas of high concentration costs the cell energy. And this uses up 1 ATP to move one molecule into the cell from an area of low concentration to an area of high concentration. A good example of this is the sugar glucose. When you eat sugar, and the sugar concentration outside the cell is lower than it is inside the cell, meaning the sugar has mostly been digested, the body or the cell wants all the sugar. And so it will continue pumping the sugar into the cell until all of the sugar outside the cell is now inside the cell. And how it does that is by an active process using a transport protein. And to move the sugar from areas of low concentration to areas of high concentration costs the cell 1 ATP molecule. Any question about any of that? All right, I think we'll talk a little bit more about that. Let's talk about simple diffusion. This is a movement of a solute from an area of high concentration to an area of low concentration. Like if you put in a drop of dye in a beaker of water, the dye is very high concentration here and low concentration there. The dye will move by diffusion from areas of high concentration to areas of low concentration. And this is the way oxygen and carbon dioxide move into and out of the cell by simple diffusion. Any question about any of that? Facilitated diffusion is simple diffusion using a protein that helps transport the molecule from one side of the membrane to another side of the membrane. Now, this molecule is moving from areas of high concentration to areas of low concentration. It's called facilitated diffusion because there's a protein helping the molecule move across the cell membrane. A good example of this is the sugar fructose, which I've already talked to you about. So whenever you eat fruit or fructose from some other source, not all of it will be digested because once the concentration of fructose will be equal outside the cell as inside the cell, the excuse me, the fructose molecule can no longer move into the cell. Is that clear to everybody?
Yes.
Great. So whenever you eat fructose, you never digest it totally. And I suppose that's a good thing. Now, I think there's this is giving you the example of glucose. And I don't want to use the example of glucose because glucose, well, it doesn't act as a facilitated diffusion, at least all the time. It's showing you this because glucose, when it is higher outside the cell than inside the cell, this protein will move glucose into the cell by facilitated diffusion, which will not cost the cell any energy. Its facilitated diffusion moves the molecule from areas of high concentration to areas of low concentration. However, when the glucose concentration equals inside the cell and outside the cell, this protein will change and it'll stop acting as a protein of facilitated diffusion. It will act as a protein of active transport. And this protein will spin ATP to move every glucose molecule outside the cell to inside the cell. Is that clear? You don't need to know that, but I'm just explaining to you why they're showing you glucose. And that is glucose, the protein that transports it, can act as facilitated diffusion, but only when the glucose is higher outside the cell than inside the cell. And then it acts as a protein engaging in active transport. So in both simple and facilitated diffusion, solutes are transported. And the transport is down a concentration gradient from areas of high concentration to areas of low concentration. Osmosis is similar, except instead of a solute moving across the membrane, it's actually water moving across the membrane. And then in simple and facilitated diffusion, they can occur across the membrane. And as we talked about, oxygen can cross the membrane. But diffusion, for example, as I gave you this example, diffusion is happening regardless of whether there's a membrane here or not. Okay? Osmosis is the net movement of water across a membrane. And it's across a selectively permeable membrane. The water moves from concentration where the concentration is high to the concentration where the water concentration is low. Does that make sense? Hopefully it does. Now, when we're talking about water concentration being high, you have to realize if the water concentration is high, that means the solute concentration is low. And if the water concentration is low, that means the solute concentration is high. Any question about any of that? Does anyone know why I'm trying to emphasize this? All right, I'm going to take that as a no. We're talking about the net movement of water. But when we're talking about osmosis and the net movement of water, we talk about it in terms of the solute, not in terms of the water. And that's why you need to know that where the water concentration is high, that means the solute concentration is low. What do I mean when we're saying that we're talking about not the water concentration, but the solute concentration? We have three different types of solutions. You can call them osmotic solutions. There's isotonic, hypotonic, and hypertonic. Isotonic is the easiest. That is where the solute concentration inside the cell is equal to the solute concentration outside the cell. So when we're talking about isotonic, you have to realize that we're talking about it from the solute concentration, not about the solvent concentration. But the solute, let's go talk about the next one, hypotonic. Oops, wrong way. Hypotonic, where the solute concentration outside the cell is lower than the solute concentration inside the cell. What happens in a hypotonic solution is that water moves into the cell. So we define the tonicity by the concentration of the solute relative to inside or outside of the cell. But it's not the solute that moves, either inside or outside the cell. It's the solvent, the water, that moves. I know that's a bit confusing, but try and understand. And the third is hypotonic, where the concentration of the solute outside the cell is higher than the concentration of the solute inside the cell. That causes the water inside the cell to leave the cell. And you can think of water simply as moving from areas where it's high to areas where it's low. The cell is happy in an isotonic solution. That means there's no net movement of water into the cell or out of the cell. And what do we mean by no net movement? That means, yeah, water molecule can move into the cell, but for every one moving in, there's one moving out. So that the water concentration in the cell doesn't increase or doesn't decrease. And the cell will be happy. You put your red blood cell in an isotonic solution, the red blood cell will be happy and will stay that way, meaning a red blood cell, for a long time. Any question about any of that? In a hypotonic solution, water moves into the cell. This will swell the plasma membrane up into the cell wall. Bacteria that have a cell wall will allow the cell membrane to expand up to the cell wall. But after that, the cell membrane cannot expand any further. That will prevent water from moving into the cell. And that's how the cell wall protects the cell from osmotic lysis in a hypotonic solution. So think about putting a bacteria cell into like a beaker of water. Water will move into the cell until the cell membrane expands up to the cell wall. After that point, no more water can move into the cell because the cell membrane cannot expand any further. And that's how the cell wall protects the cell. But your red blood cell, if you put it into a beaker of water, more and more water will move into the cell until the cell eventually pops. Excuse me. Most bacteria actually live in a hypotonic solution. And so they need their cell wall to protect them. And that's actually why penicillin G works. Because when we do penicillin G, or you take it, it damages the cell wall. And then the cell is in a hypotonic solution, and then the cell will burst. Any question about any of that? A hypertonic solution is different. The solute concentration is higher outside the cell. Think of a bacteria falling into a bottle of molasses. The water inside the cell will be higher than the water outside the cell. So water will leave the cell. That will cause the cell membrane to pull away from the wall, cell wall, and start shrinking, leading to plasmolysis. And if enough water leaves the cell, that bacteria will die, meaning generally it leads to cell death. And that's why molasses can stay on your, I don't know, cupboard or your pantry for years. And I know because my mother once had a bottle of molasses that we figured was 20 years old before I used it to make my father molasses candy, which he ate and enjoyed. And then the bottle went 10 more years before my mother decided, you know, this bottle is really old. I'm going to throw it away. And I said, you may as well, Mother. You and I haven't used it. My father was no longer alive, so there was no point in making molasses candy for him. And Mom and I didn't care for molasses candy, so we never used it. And I said, you may as well, because we're not going to use it. And we figured it was at least 30 years old, and it had never spoiled. The reason it never spoiled was because whenever the bacteria fell into the bottle, the water would leave the bacteria, and that would kill the bacteria. Any question about any of that? Okay. So the movement of material, when the material is higher outside the cell than inside the cell, can happen with a passive process. But what happens when a cell is in an environment with low nutrients concentration? For example, a cell in the open ocean. It's not going to have very much glucose or other nutrients that it's going to want in the open ocean. And the same with a cell in the desert. The nutrient concentration in the desert will be very low. So passive processes aren't going to work. We're going to need an active transport to move a molecule against its concentration gradient, because we're going to want glucose, and glucose is going to obviously be lower outside the cell than inside the cell in the open ocean or in the desert. Any question about any of that? To move a molecule against its concentration gradient is going to cost the cell to spend energy, meaning the cell needs to perform work, and that work will cost the cell energy. The cell uses an active transport protein to pump the solid molecule against its concentration gradient. The transport protein is in the cell membrane. It's generally molecule specific, meaning one transport protein moves one molecule into the cell. Like I stated, there's one transport protein to move fructose. There's another transport protein to move glucose. And those two molecules are very similar to each other. They both have the chemical formula C6H12O6. They're both sugars, etc. To move the molecule against the concentration gradient costs the cell 1 ATP. Any question about that? All right, so that's active transport, and that's found in all cells, eukaryote and prokaryotes. Prokaryotes have a specific, specific mechanism that's similar to active transport, but it's different. And let me talk about that one now. It's called group translocation, a special form of active transport. It's only found in prokaryotic cells. It does require a transport protein. It does require a high energy phosphate compound. We'll talk a little bit more about that later. And it does require, that's it. Sorry. An example is when the cell moves glucose into the cell. A prokaryotic cell can move glucose either by active transport or by group translocation. Active transport, I think you understand. So let's talk about group translocation. In group translocation, one molecule is moved into the cell against its concentration gradient. The molecule is chemically altered when it is brought into the cell. It does cost the cell energy to move the molecule into the cell. And this is where the high energy phosphate compound, such as PEP, comes into play. Meaning in this case, it's not ATP, which is very similar to a high energy phosphate compound, but it is another high energy phosphate compound. Sorry, I'm not doing too well today. I was feeling so much better, but I'm definitely sneezing and coughing. My voice is off. All right, why is the molecule changed? The molecule is changed as it's brought into the cell to help prevent the escape from the cell. It also allows the cell to accumulate more substances more easily into the cell. For example, let's say there were 10 glucose molecules outside the cell and 100 glucose molecules inside the cell. The cell is going to want to move the glucose molecules from outside the cell to inside the cell. If we were to do only group, it's not group translocation, if we were to only to use active transport, you would be pumping all 10 of the glucose molecules into the cell against its concentration gradient. And the concentration gradient is getting quite high. Everyone see that? Anyone not see that? Is there anyone there?
Yeah, sorry, I didn't hear
Yeah.
you.
Okay, great. So the point is that moving all 10 of the glucose molecules into the cell is moving against a stiff concentration gradient. If instead you move 5 of the glucose molecules into the cell by active transport, and you move 5 glucose molecules into the cell by group translocation, you have a less stiff concentration gradient to move the glucose against. With the group translocation, the glucose is brought into the cell, and then the phosphate from the high energy phosphate compound transfers the phosphate group to the glucose. And so instead of having 10 glucose molecules now inside the cell, we'll have 5 glucose molecules moved in the cell by active transport, and we'll have 5 glucose phosphate molecules into the cell that were glucose moved in by group translocation and changed into glucose phosphate. I think it's actually glucose 6-phosphate, but we won't be specific about that. Do you see why that is beneficial to the cell? Hopefully you do. Nobody's saying anything. For example, the next glucose that's being moved into the cell will only have to be pumped against 5, either 5 glucose or against 5 glucose 6-phosphate, instead of against 10 glucose. And so that's why group translocation helps out prokaryotic cells. It allows the cells to accumulate substances more easily. Any question about any of that? All right, let's move on to cytoplasm. Cytoplasm is the substance inside the cell membrane of a prokaryotic cell. It is mostly water. About 80% of the cytoplasm is water, but it does have dissolved solids, meaning all of the proteins, the amino acids, the glucose, etc., all the molecules that the cell has are dissolved in the cytoplasm. That's not entirely true. There are some molecules which will be at a concentration so great that they will not be dissolved in the cytoplasm. And I'll talk about that in a little bit. Now you should realize that in a eukaryote, it's a little different. And that is the cytoplasm in a eukaryote is the substance inside the cell membrane, but outside of the nuclear membrane. The cytoplasm does include in prokaryotes the nucleoid region, the ribosomes, and inclusions. All of these are found inside of the cytoplasm. ribosomes are so big of ribonucleic acid and protein that they do not dissolve in the cytoplasm. They're just too big to dissolve. And so they stay, what does that call you? They don't go into solution. When you come out of solution, it precipitates. Ribosomes are so big, they never go into solution. And you can actually see it here and the grains in this electron microscopic image. And I'm not seeing the inclusion. There's an inclusion there. And maybe that's an inclusion there. I don't see a mark here for an inclusion. As I stated, inclusions are areas where the concentration of the molecule has become so high that the molecules no longer stay in solution. They precipitate out of solution. And that's what an inclusion is. A region of the cytoplasm containing molecules that have so many of them that they cannot be dissolved in the cytoplasm. Any question about any of that? Shrink down. I've already talked about this. The nucleoid region is the nuclear area. We're talking about prokaryotic cells, so there is no nucleus. It's wherever the DNA is floating around in the cytoplasm. DNA in a prokaryotic cell is generally a single chromosome. There is no nuclear membrane. So the nucleoid region is wherever the DNA is floating in the cytoplasm. Any question about that? I've got it labeled here, so let me briefly state that the DNA in a prokaryotic cell does not have to be in the chromosome. The DNA in a prokaryotic cell can also be in a plasmid. A plasmid is a small molecule of DNA that's just floating around in the cytoplasm. How it's different is the chromosome has hundreds or thousands of genes. The plasmid has only a few genes. Any question about any of that? I've already talked about ribosomes. So let me just mention that ribosomes are where the cell makes proteins. So you can talk about them being the factory of the cell because at ribosomes, the cell makes proteins. You should know that ribosomes are made up of proteins and ribosomal RNA or rRNA. You should also know that prokaryotic ribosomes are smaller than eukaryotic ribosomes. And prokaryotes are 70S in size. You don't need to know about the makeup of the ribosomes. The S stands for where the ribosome sediments in an ultracentrifuge gradient, meaning the biochemists and microbiologists came up with a way to measure ribosomes. And the prokaryotic ribosomes sediment at 70S region of the ultracentrifuge gradient, which probably doesn't mean a whole lot to you, but that's okay. The the point is that the prokaryotic ribosomes are smaller than the eukaryotic ribosomes. We'll talk about the eukaryotic ribosomes later, but they're larger at 80S in size. Any question about any of that? All right, I did briefly talk about inclusions. You don't need to know any of the different examples of inclusions, but you should know that there are different types of inclusions and that they are a reserve deposit of something within the cytoplasm. Any question about any of that? So the cell was accumulating this molecules for a reason. And what happened was the cell accumulated so many molecules that the molecules precipitated out of solution, forming the inclusion. There's magnetosome inclusions, sulfur granules. The cell probably engages in sulfur metabolism, so it was packing sulfur inside the cell, and then metachromatic granules. And so much sulfur was taken in by the cells that it precipitated out of solution in the cytoplasm. All right, any question about any of that? Let's see how I'm doing here. I think I'm doing pretty good. So, of course, there's no lab today. Yeah, let me finish with the endospores. Endospores are specialized resting cells produced by certain species of bacteria, mostly members of the family Bacillus and Clostridium, create endospores. What happens is that when the environment becomes detrimental to the functioning of the vegetative parent cell, the parent cell will start making an endospore. Eventually, the environment will become so detrimental to the parent cell that the parent cell will die. But the endospore will continue. This is not reproduction because one parent vegetative cell creates one endospore. The parent cell will then die, and then we get one endospore. See, that's not reproduction. We had one parent cell, and then we end up only one endospore. So this is not reproduction. What it is a survival strategy. The endospore allows the bacteria to survive a detrimental or harsh environment. And endospores are very good or resistant to most things you throw at them. They're resistant to desiccation, heat, chemicals, even radiation. They can survive conditions that vegetative cells cannot. Sporulation is the formation of endospores. Germination is where the endospore germinates into a vegetative state cell, meaning when the cell finds a favorable environment, the endospore will germinate and convert into a vegetative cell. A vegetative cell is just the normal, growing, metabolically active cell. The endospore is not metabolically active, or if it is metabolically active, it's very low metabolism. The endospore is in a stasis, like hibernation, just waiting for the environment to change to a favorable condition, and then the endospore will germinate. And once it germinates, the vegetative cell will then go through normal metabolism, normal reproduction. Any question about any of that? Endospore formation happens similar to cell division, and that is the DNA replicates and then moves towards opposite ends of the cell. However, the cell does not divide down the middle. It divides, I don't know, at the edge or at the end. So there's one smaller cell and then the larger cell. Not cell, but division. Let's call it division. The division that's going to form the endospore is surrounded by a forespore, meaning there are two membranes around the endospore now. Peptidoglycan layer forms between the membranes. Oops, too far. The spore coat is the last thing put over the endospore. And now when the environment becomes harsh that the parent cell dies, the endospore is released, and then the coat of the endospore can survive the harsh environmental conditions, whether it's desiccation or toxic conditions. The endospore protects the cell. Any question about any of that? Come on. You'll be seeing a little video of endospore formation.
Why doesn't every home in the US have solar panels? The number one reason is not about sunlight or weather or even politics.
All right, any questions? Okay, if there's no questions, I'm going to stop here. If you have any questions on the lab, I'll be there from about 6:30 to 6:45. But after 6:45, I'm going to log off. So if you have questions about the lab, please make sure you're there from 6:30 to 6:45. There is no lab today. All right, I'll say goodbye.
Thank you.
You're welcome.